Alright, hi class. Today we're going to be talking about generating acetyl-CoA from pyruvate. And my name is Sarah Wilkins. This is Advanced Group Concepts presentation number 4. So for this assignment, in order to go from glycolysis to citric acid cycle, we must first harvest pyruvates into acetyl-CoAs. This process involves many simple steps which result in the main entrance into the citric acid cycle. In this presentation, we're going to walk through each step that is involved in generating the acetyl-CoA from pyruvate. And we're going to talk a little bit about the cofactors that we need and kind of look at how this process is reset at the end. And how that pyruvate molecule is going to bind. Okay? Alright, so first we have the pyruvate dehydrogenase complex. So if you look at the picture, we can see E1, E2, and E3. Those are essentially representing 3 different enzymes. In this complex, we have 3 distinct enzymes, and these are the enzymes that are responsible for that conversion of the pyruvate to that acetyl-CoA. And these enzymes are called decarboxylase, transacetylase, and dehydrogenase. All right, so step 1. In step 1, we have that pyruvate, and that is produced, and then it's going to be transferred over to that mitochondria, the oxidative decarbonylation, which is going to take place with the use of that pyruvate dehydrogenase enzyme. And a molecule, TPP, is going to form a bond with that pyruvate. This is, keep in mind, this is a temporary bond. Alright, now we have step number 2. In step number 2, another enzyme is going to come in here. This is called the dehydrolipoyl transacetylase. So that should sound familiar from a couple slides ago when I was showing you those different enzymes that are used here in this complex for that conversion. So this enzyme here is going to help for the other molecule, which is the lipoamide, to attach to that TPP molecule. And then here, that first step is essentially going to be removed. And by removing that, we're going to be able to form the acetyl-CoA group, which is essential. Now we are on to step number 3 here. So now that we have formed that CoA group, that group in the process is going to attack that acetyl group. And from here, it's going to form that acetyl-CoA that we've been looking for. And during this step, also, the lipoamide is released here within that step. All right, and now we're moving on to step number 4, which is the last step of this process that's going to tie everything together. So we have the lipoamide, which we talked about in the last slide. Essentially, this is going to become reionized, and it's becoming reionized with that use of the dihydrolipoyl dehydrogenase, which we also talked about in that last slide. So that's playing a role in this step as well, and helping that lipoamide become reionized. And also, the NAD is going to go on and become NADH while oxidizing that lipoamide. And you're going to be able to see that a little more on this next slide that I'm going to show you guys. So this is done so that the next cycle lipoamide can be used. So that's kind of tying that all together, and how that can go on to happen again. Alright, lastly, I just wanted to show you guys this one a little bit. You can see the pyruvate there in the beginning and then ending with the acetyl-CoA, and we just talked about the NADH, or the NAD to the NADH. So you can see how that process is kind of helping form that there. So I hope you guys enjoyed this presentation and that this helps make a little bit more sense of that process. Thanks.